Hello everyone. Today is the day. Today is finally the day that we get to complete the series titled COVID-19 from A to Zyesami. In the first video, we laid the foundations for the different terminology that is sometimes even used interchangeably in an incorrect manner in the media in discussing SARS-CoV-2 versus COVID-19 and coronavirus. We also discussed some of the relevant anatomy involved with the COVID-19 progression. In video 2, we really laid into the different mechanisms of action of different therapeutics as well as vaccines that are available. We also discussed those that have received emergency use authorization from the FDA and some of those that were waiting in the wings. Unfortunately, two of the most promising ones were not yet granted EUA. However, that is not to say that they won't get it sometime soon. A lot has changed since video 2 until now. And in this video, I intend to really highlight some of the NIH classifications on the COVID-19 progression scale, as well as talk about two of the most promising drugs, which are Humanigen's Linzolumab and Relief Therapeutic and NRXP's Aviptadil, and discuss maybe why they might not have been granted EUA just yet and what the plans seem to be going forward. Some of you have also asked me what happened to video 2B. If you'll notice, I took it down rather rapidly, and that is because someone from Humanigen reached out to me, and I have great respect for them doing so. In fact, it was in that video that I discussed a couple of topics of concern I had with the way the clinical trials were conducted or portrayed, and someone from over at Team Humanigen reached out to me, and we had the first of what's several other conversations to follow. However, after them initially reaching out to me in the beginning, I felt it was only morally correct and ethically the right thing to do that I should take down the video until further discussion, and I've had all of my questions answered. I hope that you'll see in this example, because I'm sure there'll be several others to follow, that I'm always seeking to make sure I get the most accurate and correct information out there. In doing so, there will be times sometimes that I say something that's incorrect or flat out wrong, but that's okay. The important thing is, once I realize that, or when someone reaches out to me, that I make those corrections as best I can and as immediate as I can. Now, of course, there's going to be different things that we'll have differences of opinions about, and that's okay too. However, when I re-upload the video, and anytime I do it in the future as well, I will be sure to highlight the things that I may have said that were incorrect or wrong, and tell you now the correct form of it, and I'll also show you some of the things that maybe there's a difference of opinion about for me versus the party that has gotten in touch with me. That's what it's about. Getting good information out there that is credible and that is accurate, all the while having a little fun with it. Because as you all know, on this channel we like to talk about a little of health, a little of wealth, and a little of everything else. I hope that you will continue to be a faithful and loyal subscriber after this video. And please remember to like, share, and leave a comment below on any of my videos. Alright, that's it for now. You know how we do it, so without further ado, let's get it. Okay, so let's get started. We're going to start with a little of the definitions just to do a very brief overview, okay? SARS-CoV-2 is the actual virus from the coronavirus family that can infect a person and then give you the constellation of symptoms known as COVID-19. If you get a COVID-19 diagnosis, the way that you die can be any number of different ways, usually by a secondary infection of some sort, okay? Or by COVID-19 itself going through the sequelae. Some of that sequelae is pneumonia. Pneumonia at its core simply means inflammation of the lungs. However, there's usually fluid involvement where you have fluid filling up the alveoli sacs, okay? Now, respiratory failure, there's going to be two types. You may see in some literature that there's 4 types, but there's no need to complicate things here. We're just going to discuss it as 2 types. You have hypoxia and you have hypercapnia. Hypoxia comes from the Greek word, the hypo, meaning low or decreased, and oxia means oxygen. Actually, ox means oxygen, and the IA means pertaining to what the prefix is, in this case, oxygen, but you get the picture. Anyway, it's a condition of having low levels of oxygen that you're breathing in. Now, hypercapnia, the second type of respiratory failure, means hyper, comes from the Greek, meaning increased or elevated, and then capnia, the cap part comes from the carbon dioxide, so it's elevated levels of carbon dioxide in the blood. Those are the two types of respiratory failures. Now, hypoxia, okay, that simply means that for some reason your level of oxygen that you are breathing in is low and it is not able to then infiltrate or cross that barrier and get to the blood that is circulating underneath the alveoli. Now, if you have that condition for too long, then obviously it is going to eventually lead to low levels of oxygen in the blood. At that point, you have hypoxemia, okay? So if you have hypoxemia, and by the way, hypo is Greek meaning low or decreased, and then emia is pertaining to the blood, all right? So if you have hypoxemia from a precipitating hypoxic situation, which is hypoxia, okay, then you are now diagnosed as a hypoxic hypoxemia patient. And if they test your blood levels and the carbon dioxide levels are high in your blood as well, they're not able to escape, then you are a hypoxic hypoxemia patient presenting with hypercapnia. Okay, so you can see how these things can build onto each other. Now, let's explain a little bit more what I mean by hypoxia and hypoxemia. If you are in a situation where you have thick, hardened cartilage, okay, that has replaced your surfactant in the alveoli sacs, now you're breathing oxygen in, but the oxygen cannot cross that barrier of your alveoli and go to the blood. Therefore, you have hypoxia, and then that's going to lead to hypoxemia, and then the carbon dioxide is not able to escape because it cannot escape your lungs due to the cartilage, and it cannot cross that barrier. All right, so that's why you start to present with hypercapnia as well. Now, another situation where you have hypoxemia. If somebody has sickle cell disease, for instance, in moments of stress or in high altitudes, all right, their blood takes a sickle shape, and that kind of shape is not able to carry oxygen. So therefore, they're breathing oxygen fine. They are not hypoxic. All right, the oxygen is crossing the barrier fine, all right, and goes to the blood, but the blood is not able to carry the oxygen, so now you have simply hypoxemia, but not hypoxia. And you don't have hypercapnia because the carbon dioxide is able to cross the barrier and leave the lungs as well. All right, continuing on, cytokine storm. This is a very dangerous situation, all right, that causes what I was talking about. It's going to have inflammatory proteins that can destroy the basement membrane layer of the alveoli, thus decreasing the surfactant also, or at least decreasing its functionality. And now fluids are going to come into the alveolar spaces, and it can start to harden and become cartilage. All right, that's a cartilaginous situation. Now, about 20% of patients who have COVID-19 will eventually have cytokine storm. That is the best estimates that we have right now. ARDS. ARDS is still the #1 main condition that we worry about with COVID-19. It stands for acute respiratory distress syndrome. At this stage, we are seeing sometimes only 50% of patients able to survive, and those who do survive have a decreased level of life, unfortunately. All right, acute respiratory distress syndrome, usually 50% of time is going to present with pneumonia, and also it usually is going to have some type of organ damage associated with it, and even shock. Okay, let's take a look here on the screen. So you will see here, these are tiny red blood cells known as RBCs, which stands for red blood cells. Now in each red blood cell, I want you to think of them, first of all, okay, as magic school buses. So each red blood cell is a school bus. All right. Now, on each school bus, there are going to be 4 seats, as you see these 4 pockets here. All right. So an RBC is a red blood cell. I want you to think of each RBC as a magic school bus. On each magic school bus, there are 4 seats. Ideally, you hope to have all 4 seats occupied by oxygen. This is ideally. Now, those 4 seats that you hope to occupy with oxygen, because that's oxygenated blood then, literally, all right, those seats are composed of a fabric, hemoglobin. So hemoglobin is the material, all right, that comprises or makes those 4 seats that oxygen will occupy on your school bus. One more step. Hemoglobin, the main source that you need to create the material hemoglobin is iron. So you need iron to create the material hemoglobin. Then hemoglobin is the fabric for which you make the seats. You have 4 of these seats on each school bus. This is how I want you to think about it. Now, we can talk about anemia. A means, it's Greek or Latin meaning without or lacking, and then emia for blood, all right? So when you have anemia, there's a lot of different types you can have. One type of anemia, for instance, is pernicious anemia. That's when you lack the vitamin B12 that you cannot make good red blood cells with enough hemoglobin to make 4 seats. So maybe you make only 2 seats. Why is that anemia, you may say? Because you have the same number of school buses in your blood, you just don't have enough seats. Well, because there's a certain percentage of those school buses that need to be filled with oxygen to be considered having good blood. So we can really say that anemia in this case is really having bad blood or not enough seats taken by oxygen. Now, let's look at for when a woman has their menstrual cycle or when someone is hemorrhaging inside, okay, from an internal bleed. In these cases, you are actually losing blood, so you have low blood itself. In other words, you have a low number of school buses, period, because the school buses have bled out. So when you have a low number of school buses, obviously you've lost a bunch of seats. This is anemia due to blood loss. Now, what happens in iron deficiency anemia? Iron, remember, is the main component that we need to have the material hemoglobin. Hemoglobin is the fabric that makes those seats. Therefore, if you don't have enough iron in your body, you will have anemia or bad blood or not enough seats that are taken up by oxygen. That's what I mean by there are different types of anemia. Arterial blood gases. You will notice this handy device above me or right here to my side, okay? That is a pulse oximeter, and what I said is necessary and should be essential really at every household that can afford one. They're only about $20 in a lot of places, okay? And you want to use that if you're diagnosed with COVID-19. A pulse oximeter is going to measure your SpO2 level. That's measuring the amount of saturated red blood cells at your peripheral capillaries. Capillaries is the place where oxygenated blood is going to change over from arterioles to deoxygenated blood in the venules. Arterioles are small branches of arteries. Venules are small branches of veins. Let's look at the blood flow for a second, going to what organ? Let's say the liver. So you're going to have oxygenated blood that has left from the lungs, and it is going to go into smaller branches known as arterioles, and these smaller branches is the pressure of the oxygen is beating against the blood vessels, and the oxygen will come out and deliver oxygen now to the liver. Now, since the blood, the RBCs, have lost the oxygens occupied on the seats, they will continue as blue blood now, the deoxygenated blue blood, and go through the venules, go to the veins, and then eventually come up here to the heart, where it will then be pumped to the lungs so it can pick up oxygen again to put on its seats, and you have the recycling. So periphery means like to the end of your extremities. That's why you take the pulse oximeter in your fingertips, okay? Or you can take it in the toes also, but fingertips is best. And so it's going to measure how many of those seats are occupied as the blood comes and pumps against the capillaries there in your fingertips. Now, here's the problem. It's measuring how many of those seats are taken up, but it is non-specific. So what if something else is sitting in those seats and not oxygen? This cannot tell it. And this is the dangers right now, because now with wintertime coming here in the United States, many people are starting to break the space heaters out and use those, or they're using heat at home without adequate ventilation, and that's going to build up carbon monoxide poisoning. Carbon monoxide is a serious gas that is colorless and odorless, and you can breathe it in, and the dangers is carbon monoxide acts as a bully to the seats, okay? Well, not to the seats, but it acts as a bully to oxygen to displace them out of the seats. In other words, if you have a situation where you're breathing in carbon monoxide is going to remove the oxygen and sit down in place of it and not allow oxygen to sit in those seats. And carbon monoxide and those seats are in love with each other. They have a much higher affinity than oxygen does with those seats. And when you're checking your SpO2 levels, it's not going to pick up that you have carbon monoxide in those seats. So you're going to have a falsely elevated oxygen readout on your SpO2. This is the danger of that. Now, continuing on. So SpO2 and SaO2 are really different, because SaO2 is the actual saturation of your red blood cells in your blood, and you can only take that clinically, all right? And SpO2 and SaO2 are often used interchangeably. They should not be, though, because SaO2 is a little bit more specific. And then PaO2 is going to be the partial pressure of oxygen in your blood. And when I say in the blood, I really mean in your artery environment. I know arterial blood gases gives a lot of nightmares for people in medical, any kind of medical field, from CNAs all the way up to nurses and doctors. So let's try to differentiate these a little bit and very quickly. SpO2 will measure the peripheral oxygen saturation in your peripheral capillaries. SbigAO2 is going to measure the oxygen levels in your alveolar space. The big A always stands for alveoli, and the little a always stands for your arterial environment. So SbigAO2 is checking for the oxygen in your lungs. Because if you have hypoxia and hypoxemia, and you're breathing in air, but you're in an enclosed space, like let's say that you're inside of a casket or something, or in the back of a trunk in a car, okay, you don't have adequate oxygen in your environment. Therefore, your SbigAO2 is going to be decreased, and that's the reason for your hypoxemia, okay? Now, SaO2 is going to be the arterial oxygen saturation, as I've said. PbigAO2, that's going to check the pressure of the partial pressure of oxygen that is inside of your alveolar space. PaO2 will check the partial pressure of oxygen inside your arterial space. CaO2 is going to check the actual content of oxygen. All right, it's going to see how much oxygen is truly available inside your body. PaO2 is going to check the partial pressure within your arterial environment of carbon dioxide. And then FiO2 is going to check the fraction of oxygen available in your environment, okay, in the air. And then the AA gradient is checking the difference between the partial pressure of oxygen from your alveoli space to your arterial space. In other words, it's checking to see if the hypoxemia is due to some obstruction that is in your body and not allowing the oxygen gases to exchange correctly. Something kind of cool but a little off topic is when we look at the pressure, partial pressures of oxygen, we're measuring them according to sea level barometric pressure, which is 760 mm's of mercury, or cc's. Now, when we go up in altitude, for instance, at the top of Mount Everest, let's say, for example, I believe that the partial pressure in the environment is something like 243 mm's of mercury. And so the actual percentage of that air that is oxygen is going to be the same, believe it or not, about 21%. And nitrogen will comprise approximately 79% of the air we breathe. It's not a difference in the amount of oxygen percentage in the air. The air will have the same composition, but the barometric pressure is what changes. And a lot of times people who have smoked a lot over their life or who have ARDS destruction, they lose that elasticity inside their lungs and that surfactant inside their alveoli, all right, that is going to have the proper surface tension for gas exchanges. Those things collapse, and now you do not have that elasticity to breathe because it's actually like the negative vacuum space in your lungs that causes you to inspire. This is why we have different types of ventilation settings, including positive pressure ventilation, where it's actually pushing the air into your lungs, okay? And so when we have certain sports teams, let's say the Atlanta Braves, for instance, our baseball team, and also Atlanta Falcons, our American football team, when they go up to play a team like the Colorado Rockies or the Denver Broncos that are in a much higher altitude, they're not accustomed to having that little barometric pressure, and therefore you will see them on the sidelines often trying to gasp for air and get oxygen in them. And conversely, when we have the sports players come from higher altitudes of training and they come down to sea level, it's like they have much more endurance and stamina. Let's talk about a few challenges that have presented themselves during this COVID-19 pandemic. One of the challenges has been the variance of statistics. In resource-limited settings, in-hospital mortality rates may be higher than those reported elsewhere. Not because the COVID-19 is actually higher, but because the resources could be limited. Therefore, the hospital does not have the capabilities to take care of the patients as well. Therefore, more patients die, causing a higher mortality rate, or it's possible that they just don't have the staff available. That counts as lower or limited resources. One example that they did with 10 different countries in Africa, it showed that all 10 countries had a median, for instance, of 2 intensive care specialists at each hospital, but a small amount of facilities in these countries did not even have a pulse oximeter. They lacked a pulse oximeter. The thing that we just looked at a few slides ago that I said was essential for every home to have if possible when someone is diagnosed with COVID-19. Now, we saw mortality rates associated in these hospitals going over 50% sometimes, but this is not consistent with the mortality rate of people who have COVID-19, as you know. And so this is what I mean. There's such a variance of statistics. It's very hard to get our hands on. For instance, one limited statistic we have is how many patients are going to actually progress to ARDS, how many patients are going to stay at the beginning stages of COVID-19 and have asymptomatic COVID-19, because we're finding now that different statisticians and different hospitals, once someone is diagnosed with asymptomatic COVID-19, they did not follow up to see if that patient ever got symptoms eventually anyway within the next few days. And some symptoms, remember, can take up to 14 days to show. This is why there's a scientific basis for the 14-day quarantine, because sometimes it takes up to that for symptoms to actually manifest. But regardless, mortality is always associated with underlying comorbidities as well as resource shortages. This seems to be consistent, and that comes from UpToDate.com, which is very credible as a source. When it comes to equity of healthcare, minorities are still among the highest infected, and this especially is speaking about, at least here in the United States, blacks and Latinos. They are amongst the highest infected, and it's due a lot of times to limited resources, previous comorbidities, many times that are undiagnosed, and inadequacy of previous health care in general. It's not a genetic or hereditary predisposition that minorities have that causes them to get COVID-19 more, but it is these other things that I've just named. In other words, it's a heavy influence of the social determinants of health. And we really are seeking to find an equitable solution during these times. The final challenge I want to talk about is an acceleration of COVID-19 therapeutic interventions and vaccines. With limited resources, there's a great need to coordinate and streamline processes to make the best use of biomedical research and resources and testing of preclinical compounds. And so there's been a couple of solutions that seem to have arisen during these times. Possible solutions that have arisen so far, one of them is the active trials, and that is under the NIH umbrella, as we know, specifically the Allergy and Immunology division of NIH. And then let's skip down to NCATS there. That is another division of the NIH, and they are going to use computerized algorithms to try to match up pre-approved drugs that we already have approval for from the FDA and see which ones have the highest chance of actually working against a particular virus. And it will run that scenario out or do a trial of that scenario on computer that simulates real life. The problem with that is we found things that look great in vitro, but then when it's in vivo, it does not hold up. Remember, I talked about hydroxychloroquine fell into this chasm, and it looks great in vitro. But when they used it in vivo, it was causing phospholipidosis at extremely alarming levels. Now, let's look at quantum leap healthcare. This is a great solution that has come and was birthed out of the Silicon Valley of California. Many great things come from Silicon Valley. Elon Musk, for instance, if you will. Now, of course, I know he's really from South Africa, but we'll claim him. He was living in Silicon Valley when he helped create eBay and PayPal, so you get where I'm going with it. Anyway, Quantum Leap Healthcare has an extensive amount of resources and a great broad network and plethora of hospitals and academics that works with it. These are the people who are responsible for the I Spy trial that Aviptidil, the inhaled form, is currently enrolled in. Okay, let's discuss the NIH classification categories of COVID-19. First, we have asymptomatic/pre-symptomatic. There's no real concise and consistent agreement about how many people diagnosed with COVID-19 actually belong in this category. However, 30% seems to be the best estimate. As I've said, how many progress on to severe COVID-19? It seems to be 20%. And how many of that 20% will actually get cytokine storm? That seems to be 20% again, at least according to the Washington University St. Louis study, which is the only study to my knowledge that has been done for cytokine storm. This is one reason I was saying about Humanigen and Linzolumab. I love the drug. I think that it's going to be a huge success, but people were starting to look at it and blow it out of proportion. This is going to be 20% of 20% that it's going to apply to, most likely. And with that being said, I am not criticizing it. I'm just saying we need to make sure we are looking at things in their proper context. That includes for many investors in Aviptidil, we have to look at things in their proper context, because sometimes people can pump something up so much that it causes a lot of damage in extenuating circumstances downstream that they may not even know. Now, back to this. So when we go on and we look then at the next categorization, which is mild COVID-19, patients that present with mild illness may exhibit a different variety of signs and symptoms like fever, cough, headache, the pathognomonic loss of taste and smell, nausea and vomiting. All right, those things are going to exist at the mild level. However, what will distinguish a patient for being mild and not yet in the categorization of moderate COVID-19 is going to be that patients in the mild category do not have shortness of breath or dyspnea with exertion. Dyspnea is going to come from the Greek or Latin dys, dys meaning difficulty, all right, like dysfunction is difficult functionality, and then pnea, P-N-E-A, is coming from the Greek with breathing, all right? So this is going to be that someone with exertion is having difficulties in breathing. That's a little different than actual shortness of breath, all right? Now, continuing on, patients here in mild also will not have abnormal imaging, all right, on a CT scan or chest x-ray. When you have shortness of breath or dyspnea with exertion or abnormal imaging, that is going to then put you on into the moderate category, okay? This is one of the big things that's going to separate these classifications of patients. Once you go to the moderate category now, the big thing to watch out for is what's going to separate moderate and severe. Moderate patients, in those patients, there's evidence of lower respiratory disease during the clinical assessment or during the imaging, as I've said. But these patients still have an SpO2 above 94% on room air at sea level. If that drops below 94%, now we're getting to the severe level, okay? Once again, if it drops below 94%, now we're getting to the severe level. If bacterial pneumonia or sepsis is suspected at this stage, this is the time to administer empiric antibiotic treatments and reevaluate the patient daily, okay? You really have to watch them at this stage between going from moderate to severe. Let's look at the severe illnesses. What happens in this stage? Well, as I've said, you're going to have less than 94% of oxygen on room air at sea level. Also, the PaO2 divided by the fraction of oxygen, all right, is going to be less than 300 mm's of mercury. And also, they may present at this stage with respiratory rates greater than 30 breaths a minute or with lung infiltrates greater than 50%. At this stage, you're definitely going to see the hypoxic hypoxemic patient probably presenting with hypercapnia as well, as we have discussed. Now, unfortunately, the next stage is not good, and this is the critical illness stage. At this stage, you have full-blown ARDS many times. Septic shock may be presenting. Also, you may have an exacerbation of underlying comorbidities right here. And this is not good. This is the stage where we're seeing sometimes upwards of 50% of patients, unfortunately, not making it. Another classification that NIH has, which I'm glad they created, was the special populations category, because there is very little research on patients in this category. That's going to include your pregnant patients, your children, patients with cancers, patients with influenza, patients with HIV or transplant patients as well. Now, this is one part that I really love the drug Aviptadil again, because it really shines, because it has already worked on patients in past former trials that have had transplants. In fact, transplant lung surgeries. So that's really amazing. Okay, so let's talk about the drug development process at this point. A lot of people have asked about that, and there has not been a lot of transparency, to be honest with you, at least in the past. Hopefully those kind of things are changing now. When we look at the drug development process, you're going to have people to break it up in different ways. One thing that I would say is pretty consistent is that during the research and development phase as a whole, it is typically around 10 years, and then the marketing phase, where it's actually post-marketing really, and the drug is out there, is about another 10 years. So the overall drug life cycle tends to be consistently around, I would say, 20 years. Okay? And for the research and development phase, the actual research, you can break that down further. For instance, doing the actual research is around 5 years, and then the development of the drug can take another 2 to 3 years, all the way up to 7 years, to be honest. And then post-marketing, you're going to have, as I said, around 10 years. It depends on the drug, obviously, and it depends on what the FDA says. The FDA, we can really say, directly affects how long a drug takes to get out there to the people. Now, that may seem like a controversial statement, but I'm not meaning it in a critical way. I'm simply saying that the FDA is the gatekeeper. So, of course, they directly affect how long it would take for the drug to get out there to the people. And the FDA has their own reasons for validating a drug or denying it, saying whether it is safe and may be effective during times like right now where drugs are asking for an EUA, such as linezolizumab from Humanigen and Aviptidil from Relief Therapeutics and NRXP. So let's talk about this drug development process. The research and development phase, as we talk about it here, and as the FDA considers it, that's going to be done before the preclinical research. If you talk to an investor, for instance, or let's say some of the sponsors who are looking to make the money from the drug, they will call the research and development phase anything that is pre-registration. And registration is the last part of the FDA process when you actually get the marketing license for the drug. So it depends on who you ask what R&D is talking about specifically. In my case here, I am specifically looking at it as the FDA looks at it. They are going to call the research and development phase everything preclinical. At the preclinical research phase, this is when you're going to test the drug in vitro so that you can prepare it for in vivo. This is where things like NCATs is really coming in handy. That's the NCATS that I showed you earlier, a division under the NIH, because they are actually able to run different simulations and at least give the best possible scenarios, okay, to some of these companies so they don't waste so much money, and more importantly, so they don't waste precious moments from people who are really needing this drug. Now, stage 3 is the clinical research. At this phase, this is when you're going to have the different phases of 1, 2, 3, and 4, and seeing the efficacy of the actual drug. Stage 4 is going to be the FDA reviewing the drug. This is when you're going to have the NDA to be submitted, and the FDA is going to review that, and hopefully, or at least the sponsors are hoping that the FDA approves the drug. The FDA at this stage may call in different advisory committees to help them on making some decisions. And finally, step 5 is the post-market safety monitoring phase, when it is out there in the world and the FDA is continuing to monitor the drug for safety. One thing you will notice is consistent in all of the research processes and in the processes of drug development is safety first. Okay, now let's go to the clinical research phases. In phase 1, the purpose of this phase is safety and dosing. Usually, you're typically going to have 20 to 100 healthy patients with the disease or condition. All right, the duration is usually several months, and 70% of drugs succeed past this. Phase 2. Again, I'm going from the FDA's perspective and point of view. The purpose of phase 2 is for efficacy and side effects. Usually you will have up to 100 people with the disease or the condition. The duration again is about several months, but could go up to 2 years. Approximately 30% of drugs succeed. Now, here's one reason why I did not think the EUA passed for a Viptidil is because there was only 130 or 140 patients for them to review, whereas phase 2 typically is going to have up to several hundred people with the disease or condition. Now, I know some people are going to say, what about the EAP and what about some of those other trials that they've read the great news on, but those trials were not RCTs, random controlled trials. And this is what the FDA has stressed they really want, even in times of EUA. Now, phase 3, the purpose of this is efficacy and adverse reactions. Okay, and so here you're going to need 300 to 3,000 patients typically with the disease or condition. The duration of this stage is typically 1 to 4 years, and again, 30% of drugs succeed past here. And finally, you have stage 4. The purpose of this is safety and efficacy again, and you're going to have several thousand patients with the disease or condition, and this part is going to be ongoing, constantly monitoring, constantly looking. It does not take much research to look in the news and see drugs that have been out there for a long time that were deemed originally safe for administering, but suddenly the FDA will pull it because new information has become apparent. Let's talk a little bit about the two companies that this whole video is kind of hinting towards. The name of this video was originally, as you will remember, 3 companies, 2 drugs, 1 EUA, and how it changed the process forever, or changed COVID-19 forever. Unfortunately, I had to rename it, how it may change COVID-19 forever. The reason being, quite frankly, the FDA did not give either of these drugs approval, those drugs being Viptidil and lislizumab, as I thought they would by now. The FDA has surprised many in some of the somewhat seeming inconsistencies that has happened since COVID-19 began. When we look at Humanigen, the reason why the FDA denied it was because it was unable to conclude that the well-known and potential benefits of lislizumab outweighed known and potential risks, or use it as a treatment for COVID-19 because of it. So in other words, whether there was not enough data or whether the data did not show safety enough for them, the FDA chose to decline to approve it. Furthermore, lislizumab being in the category of GM-CSFs, the FDA is looking at it as a hodgepodge of multiple drugs that function and act the same way, as if they expect all of them to perform the same way, which is just simply not true. Let's look at the GM-CSF done by GlaxoSmithKline, excuse me. Their trials failed to meet statistical significance, but they included mechanical ventilator patients. However, the Humanigen trials did not include mechanical ventilator patients. Maybe there's something to look at there that, GM-CSFs are effective on sick patients, but not too sick. So now you start getting into the nitty-gritty of what we went over in this video and dividing up the differences between, moderate and severe and critical patients, okay? That's why we went over that, because all of those things play a part when you start talking about these kind of things. Furthermore, they were considered, or the FDA considered that in regarding the special patients, remember that 6th category there that we went over? In those kind of patients, they were concerned that GM-CSFs, especially like cancer patients or transplant patients, that it would just put them at too much risk. However, that has not been proven in any of the trials yet. Nevertheless, as I've said many times, in the right and proper context, Linzolumab has its place, and I really do hope that it gets approved. Furthermore, that right in proper context is approximately 4% of the patients with COVID-19. As I've said, 20% of the 20%, because 20% of patients who will get COVID-19 will go on to the severe stages. Then 20% of them will consequently become, at the stage where they have cytokine storms. That's consistent with what the Washington University of St. Louis has found in saying that about 4% of patients with COVID-19 actually exhibited the levels high enough of the inflammatory proteins to show that they were in cytokine storm levels. Therefore, the utility of linzolumab is going to be for about 4% of the COVID-19 patients. Furthermore, there was also only one real study done for Humanage's linzolumab, and there was a lot of questions about that. 2 other studies they were going to do had to be stopped for various reasons, and currently there are 2 active trials that are going on right now. The one that they did themselves is completed and has been published, but the other 2 trials, one is for the C-SMART platform in Australia, and the other one is going to be part of that active group of trials that's going on here in the United States. Nevertheless, linzolumab has been tested on at least 243 patients that the FDA was able to look at and conclude their decision upon, versus Aviptidil, which has only had 130 patients that the FDA was able to actually conclude the safety data on for the RCT that was done by the group NRXP. The 3 companies I'm discussing are RLFT, which is Relief Therapeutics, RLFTF, and NRXP, which is the NeuroRx Pharmaceuticals group. They are the partners for Aviptidil. Now we go over here to Humanagen, and they are the ones who are over linzolumab. So those are the 3 companies. The 2 drugs are obviously linzolumab and Aviptidil. 1 EUA, because there has still been only 1 EUA really for therapeutics at the latter stages for someone with COVID-19, and that does not go too far into the latter stages. It doesn't cover severe or critical or even moderate, alright, and that's remdesivir by Gilead. Alright, that's the only EUA that's been doled out for therapeutics like that. Now, Acterma, also by Roche Holdings, it was recently granted approval. It is a MAB, so you can see how the ever-changing face of things is going. Remdesivir still sets that standard to which Aviptidil is measured against. Aviptidil currently, they have 5 active trials. They have 1 trial that was completed, and they have the EAP, which is the the Expanded Access Protocol that is currently in place as well. Now with Aviptidil, unfortunately, only 134 of the patients out of that 196 patient group for the first trial was able to be observed for their safety and efficacy. Dr. Javid from NRXP is currently trying to pull some of the other patients from the actively running active trial, the active 3B if you want to look it up, and try to compile to where there's at least 150 to observe. The only problem I see with that is, if you remember when I said the different phases of FDA, well, Aviptidil is in phase 2B/3. They're doing a combination of the phases so they can look at the safety and efficacy as well as the dosage, and also look at any adverse side events, okay? But typically for a phase 2 trial, you're going to have several hundred patients. 134 or even 150 is not several hundred. When we get to the phase 3 part, the FDA generally likes to see between 300 up to 3,000 or 4,000 patients, okay? Remdesivir checked all of these marks at their various stages, and that's one reason it was approved so quickly. Plus, they had a lot of data in the background that they were waiting to just dole out, because they've tried remdesivir before, but unfortunately it failed back when we had the SARS-CoV-1 infections and the MERS-CoV scare. Okay, so with Aviptidil, this is why I say I think it's going to have to wait until the NDA, go for the full drug approval process, because I just don't see where even 150 patients is going to check those marks. Now I know we're looking at EUA, Emergency Use Authorization, And the FDA has already proven that different things and different measures can be taken during this time. For instance, I think that they're fine with approving GMP processes as opposed to cGMP processes, which have to be approved during the NDA full approval process for most drugs, okay? But still, we are seeing the FDA be a little more cautious, because please remember that it wasn't too long that there was a big debacle about remdesivir. The jury is still out about whether it's really that effective or not, okay? The World Health Organization, actually took the opposite stance and advised against giving approval to remdesivir based on insufficient data. However, the FDA chose to still approve it, okay? And so this is what we're seeing when I talk about they are the gatekeepers and the inconsistencies that exist out there. When the FDA approved it, who was against it? EMA continued to kind of deliberate. They finally approved it, all right? And so now when you talk about a drug like Aviptidil, that we've seen the great progress of these patients that have taken it, how it's reversed even the damage in the lungs. We've seen the x-rays, but for FDA standards, they've seen 134 patients, basically. Now, when it comes to Humanigen, there's only one clinical trial. That's just simply not enough. There needs to be more evidence. As I've said, there are 2 more that are open right now, one in Australia, one with the active group here in the United States for Humanigen, for the linzolumab drug. When it comes to Aviptidil, there are 5 active trials right now. You have the one that was completed, then you have the EAP protocol, and now you have 5 that are actively recruiting for both a mixture of the inhaled form as well as the IV form. But never to have a shortage of drama in the biopharma world, NRXP and RLFTF are currently at odds with each other over the collaboration agreement and whether it has been breached or not, as well as many other accusations that are currently being played out publicly and may eventually lead to a court case, but more than likely it will probably lead to just arbitration, in my opinion. If you want to know more about that, then please look at my video going over the court cases. Now, again, just to remind everybody, the juicy stuff, the juicy details that I have been promising in video 2A, they're still there in video 2B. It's set as private right now, but I will be releasing that at a later date. Just let me get through these phone conversations, okay, so I can make sure that I'm giving accurate and credible information. That's all I've got for now. I hope that you'll continue to be a faithful and loyal subscriber. I hope that you will also like, share, and even leave a comment on this video below, as well as many of my other videos, okay? And I look forward to talking to you more. This video series was mainly to educate people about COVID-19 and to bring into play where do the 2 drugs that have seemed to get a lot of recognition and a lot of fanfare about them, linzolumab and Aviptidil, where they would insert into that process. So now you see Aviptidil will insert there and cover, the moderate, the severe, and critical patients. Although the first EUA was done just for the critical patients, but you can see them now trying the inhaled version at an earlier stage of that COVID-19 progression, I should say. For Humanigen, you see which stage they will cover, which is that specific cytokine storm effect that could happen from that release of the pro-inflammatory proteins. Alright, I hope that I've helped, and I'll see you next time.